welcome to another screencast where I'll be discussing genetic variation and sort of the sources of genetic variation. So by the end of this, hopefully you're able to describe and explain, number one here, the evolutionary significance of genetic variation. So like this is answering the big picture question, kind of who cares, right? So what does it matter? And then number B here, sources of variation at the DNA level, right? And so I'm going to consider this at the DNA level, and then we'll consider this sort of at the chromosome level. And I think I'll split this into two screencasts. So I'll deal with this C in another one. I think it'll take me too long to kind of get through all this. But so sources of variation, right? How does DNA replication generate variation? Mutation, transformation. So this is a new thing. Transduction is a new thing. Conjugation is a new thing for you. Transposition is a new thing, and this recombination is kind of sort of new, right? So let's focus there, and then ultimately, right, how each of the processes above produces and maintains genetic, that is DNA sequence variation within populations of organisms. Right, so the evolutionary significance of this is that the genotypic variation, and I'm just reading the title up here, right? The genotypic variation can cause phenotypic variation. So if I vary the genotype, I can vary the phenotype. If I vary the phenotype of the individual organisms in the population can alter survivorship. That is, some are able to survive, some are not able to survive, right? So that means then that genotypic variation can alter survivorship, right? So if A equals B and B equals C, then A equals C, right? So if I generate some genetic variation, I can then alter some sort of survivorship of individuals in the population. We'd seen that with our lab, right, where we had this recombinant chromosome, this recombinant plasmid here, right? And then this is this was E. coli, right? And so this E. coli does not contain this gene right here, which was AMP-R, which is the resistance for ampicillin. Right? It's coded for the resistance to ampicillin. So these E. coli in the environment of ampicillin, so if I put these E. coli in an environment where there is ampicillin, right, these E. coli do not survive because they do not possess this gene. Their genotype here is that they don't have it. But if I transform these E. coli and get them to take up this plasmid right here, now these E. coli have changed their genotype, right? They actually have this gene now as compared to the E. coli over here that do not have the gene. And now this genotype change conveys a phenotype change, the phenotype being that they produce enzymes that break down ampicillin, and now they are able to survive in that ampicillin environment and then reproduce, that is, make more of themselves. And when they do that, they reproduce this AMP-R, right? And then all their offspring then inherit that AMP-R. And so you have now changed the phenotype of this population. Similar on the other side here is that farmers are spraying these pesticides on their fields, and you can assume that there is some variation within the population, right? So if there is this variation in the population, so these are the little pests in there, right? And each one of these represents, times whatever, thousands, right, of individuals in that population that possess this variation, right? So here, some of these individuals right here, for one reason or another, like the source of that's the rest of this PowerPoint here. The source of this variation is what we're getting at, but the fact that this chromosome with a gene conferring resistance to insecticide, right, this is a different sequence than the sequences that are on these individuals over here. So this genetic variation conveys a different phenotype, right? This is the phenotype, right? Resistance to the insecticide. So who gets to survive? The individuals in the population that possess this, right? So this survivor then mates with other survivors, right? And you then end up getting and breeding a population of resistant insects, right? And so now we say that this population of insects has evolved, right, with this selection pressure. So this is a natural this is like an artificial selection kind of, but it's a natural selection, in that the variation existed already within the population. Of course, if this variation did not exist in this population and the population was homogeneous, right, everybody had this, then yeah, right, that insecticide is very worthwhile and effective, right? The same thing goes over here with the ampicillin, right? If all the E. coli are not resistant to ampicillin, then the ampicillin, the antibiotic, works. But all it takes is one or a handful of individuals to possess some sort of variation within a population, and now this population stands a better chance of surviving and evolving. Right? So you can click that animation over here, right, to watch that. So what are the sources of the variation? The rest of this is dedicated to again, identifying the different sources of variation. So we go back here, we're focusing right over on these guys here. So one source of replication is that DNA replication is imperfect, right? Most times mistakes get fixed, right, when you're doing the replication, but sometimes they don't. So DNA replication is one source of this variation that might creep in there, right? It's not a perfect system, right? It's pretty close, but it's not perfect when you're replicating. Number two, genotypes, changes in these genotypes could be due to mutations, right? Single nucleotide polymorphisms, right? So you have mutagens that can cause this, right? And sometimes just in the replication, you get a different nucleotide base added in there, right? That replication can be a cause here of this. Mutagens, right? Things that are causing nucleotides. Radiation is an example. Radiation can be an example of that. So mutations, we've talked about those previously. We want to focus on a few different ones here, specifically how viruses can affect the genotype of specifically prokaryotes, right? So the rest of this variation, these variation slides are pretty much dedicated to getting variation within prokaryotes. This is a big deal because prokaryotes, you think, all right, well, they just reproduce asexually, right? So if there's asexual reproduction, you know, via some sort of binary fission thing, that you're just copying, right, and it shouldn't be a problem. But there is variation in prokaryotic populations. The genomes are different. The genotypes are different. But how does that arise? Right? First of all, if we go back and look at viruses here, right, we said that they are sloppy intracellular parasites. So this replication process here, right, and this reverse transcription process is not as neat and tidy, right? So there's a lot of spelling mistakes that get made here. And so viruses can evolve their genomes very relatively easily, and some viruses are more notorious than others for having little mistakes there and generating variation within their genomes. Right? HIV is a pretty highly mutatable, mistake-laden genome. The lytic and lysogenic cycles lead to the production of more viruses and potential to recombine genetic material. So what we're going to say here is that now let's focus on these prokaryotes, right? So how am I going to get these prokaryotes varied? Well, we notice that right here, this lysogenic cycle, remember that the viral DNA can become incorporated into the host DNA right there. And then it comes out of the prophage, right? And then gets into other ones. If this is unfamiliar to you, go back and watch the screencast on viruses. Right, so in another source of variation generation, transduction. This process of transduction is transmission of genetic information between prokaryotes via a viral vector, right? So the diagram shows you what's happening. So let's step through the diagram here. So in transduction, you're using a virus to move genetic material, a gene or an allele from one cell to the next, right? So this is bacterial cell A, let's call this. So here's bacterial cell A. And this is the phage, right? So it's going to inject its DNA into there. And this is indicating that you have two genes, right? There's an A gene, and then there's a B gene, and you have this plus allele, and then the B plus allele. So two genes, an A gene and a B gene, and then two alleles, and these are at two different loci, right? So this is on the chromosome of this host cell. Now what happens is, as the viral DNA is injected, remember that the host DNA is sort of chopped up, right? It's hydrolyzed into pieces over here, right? And then you use the host cell machinery to generate more viruses, right? And so you get more capsids, and then the viral DNA gets incorporated into the capsid here. But this is the point of transduction right here, is that sometimes, right, this has been known to happen, this has been shown to happen, that the virus didn't pick up viral DNA, right? Here the virus picks up, or just happened to pick up the A plus allele, right, from this hydrolyzed DNA up here. Right? And now this virus doesn't know any better, doesn't know any different, right? But it will now go and infect. This is E. coli B, right? Actually, we should just call this 1, right? So let's call this number 1, and this is cell number 2, right? And so the assumption here with cell number 2 is that it also has an A gene and a B, sorry, an A gene and a B gene, but now the allele types here are different, right? So this has an A minus and this is a B minus, and again the plus and minus are just to indicate the two different alleles here. So what happens now is that this virus injects the DNA, right, from this cell number 1 into cell number 2, and the cell number 2's DNA is not all hydrolyzed and cut up because it doesn't have the viral DNA. Remember, this viral DNA is leading to the signal to sort of hydrolyze all of the host DNA right there. But that doesn't happen here. But instead, what does happen is that this A plus allele and the A minus allele here match up, and what you have then is essentially a crossing over, right, or a recombination, right? So this process right here, where you're switching the plus and the minus alleles, generates a recombinant DNA. So now this is a recombinant bacterium. Right? And so if you read this, the recombinants have genotypes A plus B minus, different from either the donor, which is A plus B plus, or the recipient A minus B minus. So go get back up here again. So the genotype of this was A plus B plus, right? The genotype of cell number 2 was A minus, B minus, and now the recombinant one is A plus and B minus. This is This is a method of generating variation. Right? So click on the animation over here to watch and review that happening. Right? That's the third source. Another source of variation at the prokaryotic level, at the DNA level, is conjugation. Right? So conjugation is a direct cell-to-cell transfer of genetic information between prokaryotes. This is the closest thing that prokaryotes would have to a sexual reproduction, right? That involves, two distinct cells here, right? And so this is what's going on. We have an E. coli cell here, say, and here's an E. coli over here, right? And you notice that this E. coli has this plasmid in it, and this E. coli does not. Right? And so on this plasmid, there are genes that, when they are expressed, code for this sex pilus thing, right? And this is also sometimes referred to, or I've seen it referred to as a mating bridge, right? And now this plasmid also just happens to have antibiotic resistance genes on it. Many antibiotic resistance genes are on plasmids. And so this one happens to also have that. And so what happens is, when the genes on this plasmid get expressed, this bacterium forms this mating bridge, or this sex pilus here, which then goes over and docks and connects with this E. coli over here. Right? You get these conjugative proteins, which are also coded for right here. And now what's happening is that this plasmid is being replicated, right? So conjugative, right? Changing, it's replicating this plasmid, and now the plasmid that's being replicated is actually moving into this other E. coli cell here. Right? And then the cells separate, and now you've essentially changed this E. coli from a minus E. coli now into a plus E. coli, plus indicating that it has this plasmid. And remember that this plasmid contained an epsilon resistance gene, so now I've changed the genotype of this E. coli. I've changed its phenotype, right? And that can convey survivorship differences. So again, this is a way of varying the genome of this cell right here. So there's a couple of animations to take a look at. I think that was 4. This is what? This is number 5 here. Transformation. You should be very familiar with transformation. We did this lab, right? So transformation is another way to get variation. And this is defined as the uptake of naked DNA. Everybody laughs when I say naked DNA, right? But meaning that it's not in a cell, right? Not in a cell. Now, this happens because cells die and rupture, and DNA is fragmented and hydrolyzed, and that stuff in the environment sometimes is hanging out in the environment there. So these are DNA fragments from donor cells. Could have been a dead cell, could have been a cell that was hydrolyzed or lysed or something. So here is the organism here, and its genotype is little a, little b, little c, little d, little e. Right? So there's an A locus, a B locus, a C locus, a D locus, and an E locus. And this happens to have the lowercase alleles at all of them. Right? But the DNA fragments from the donor over here also have A, B, C, D, E loci on them, but the alleles are different. Right? So these are the capital letters. Right? So what happens is just randomly the recipient takes up DNA, this donor DNA, and now I've got, in this example, they took up the A and a fragment that has the A allele and that has the A and the B locus on it. And these alleles happen to be different. And so what's going to happen here, is that one of them, this B one, happens to recombine right there. And so now I've changed the genotype of this cell down here to be little a, big B, little c, little d, little e, right? And here's variation. This is another way that prokaryotes can vary their genomes. Right? And so, again, click on that animation to watch that process a little bit more. Okay. 6th, transposition. Transposition involves the movement of DNA segments within and between DNA molecules in prokaryotes and eukaryotes. So now those previous ones are primarily prokaryotic. Now we have here this eukaryotic added in here as well. Transposition, position, it's changing the position, right? And so we have 2 methods of this transposition, right? 2 methods here. One, cut and paste mechanism, right? Where the segment that is going to be moved, right, is called a transposon, and then here's the target site. So literally cut and paste, right? I'm going to cut DNA, right, with some sort of nuclease, right? And then I would cut with a nuclease here, right? And then this transposon gets moved from this part of the genome to this part of the genome, and then we use a ligase to seal that up. And now this is a recombinant DNA right here. Right? And so I've moved this gene from this location to this location. Right? Now, notice that it got put in between these purple areas, so now this maybe interrupts the expression of this gene. Normally, it'll change the expression product here. So again, I've now changed the genotype at that sequence, but this is like in-house moving. So this is from one area of the genome to the other. This isn't bringing it from another cell or something like that. Right? Another mechanism is the copy and paste mechanism. So this is cut and paste. This is copy and paste. So here's our transposon again. Right? We make a copy of that and then move the copy. Right? So you keep this in place and then you move the copy over there. So I've changed the genome actually in 2 spots, right? I changed it there at A, and I changed it B here. Right? Here, I've just changed it at this B section. Right? So transposons, this is another way of generating variation. Right? And again, there's another animation to watch. Okay. Lastly, maybe 8, genetic variation can be generated in a gamete or a spore via recombination, that is crossing over between homologous non-sister chromatin during meiosis, right? So again, now this is meiosis, so this is strictly a eukaryotic mechanism. But this is, you should see a similar theme here, right? Is that I've got this happening here, right, where I'm going to be exchanging segments of DNA, right? So I have to cut DNA, I have to move it, right? Seal it back up. So this is this is this recombination that happened with the transduction. It happened with the transformation. It happened with the transposons, right? So what you should do now is kind of stop and pause here. And what is similar and what is different between these variation mechanisms, right? So generate yourself a thing right here. And so we have, 7 of these mechanisms, right? 1, 2, 3, 4, 5, 6, 7, right? What's similar between all of them, but then what is different between them, right? All. So you should be able to identify those, describe what's similar and what's different, right? So, like I said, I'm going to stop there, and hopefully you should be able to now describe, at this level right there. Okay. Thanks for listening, and check out the second half of this.